morning, viewer. Welcome to our Globe TV as a breaking news for viewer. Yeah, studying the process of brain formation illuminates just how much of development is a series of teeny miracles. Yeah, only a few weeks after a human egg is fertilized, a set of cells called the neural play widens, stretches, and rolls away to create a tube. So this delicate dance of cells forms that will become our brain and spinal cord, the basis of our thoughts, feelings and actions. How the cells know what they will become is not well understood till now. But a new research by Eric Brooks, a developmental biologist at the NC State College of Veterinary Medicine, and his colleagues offers clues. By studying how RNA expressions pattern change across cells over time in a very early mouse embryos, the scientists developed a map of the neural plate and followed it through time as it formed the neural tube. In the process, Brooks and his colleagues showed that they can pinpoint where a cell will move in the early brain based entirely on the RNA that cell is expressing. Published in eLive, the findings could help scientists around the world as they work to understand brain development and how to stop that development from going wrong. All embryos start as a ball of cells. Those cells multiply and differentiate over time into the tissues we recognize as our muscles, bones, brain, and organs. The cells that will become the central nervous system, the brain, and spinal cord are some of the first to get into formation. Brooks has always been curious to understand how the cells know what to go, how they understand what their eventual place and role will be. A lot of people are fundamentally interested in where the brain comes from, Brooks says. Scientists know that chemical signals from gradients, concentrations that help guide cells to where they need to be, but the changes happening inside those cells, changes that will make those cells into different types of neurons and support cells, and all interacting with one another in highly specific ways remain a mysterious dance. What is it that's really helping to differentiate these cells from one another? Brooks asks, or allow them to do specific things like create the shape of the issue? The complicated choreography offers many opportunities for missteps. Defects in the formation of this structure, the cranial neural tube or the neural tube more broadly, are among the most common developmental defects in humans. Brooks says affecting one in every 2000 birds. May the exclusive report Glory Globe to be Delhi Bharat.